Hello and welcome to Pharmacy Skills. Many of us healthcare professionals, unless you specifically studied microbiology, only had a module on microbiology in one of our years. So it's easy for us to forget the fundamentals on microbiology when revising anti-infective medications and infections. Studying infections often uses many microbiology terms that if you don't understand the basics, you might easily get lost. So this video is a pre-infection video covering everything you must know before we dive into the infections and anti-infectives. If you're watching this on YouTube, please consider visiting us at home at pharmacyskills.com. Microbiology is the study of very small living things we call microorganisms, things like bacteria, fungi, protozoa, and even includes viruses. Although viruses themselves aren't exactly living organisms, they're a special type of trouble, and we will get into that shortly. These organisms just want to survive and replicate, just like us, just like any other living thing, which is fine, except when they do it inside of you and start to cause trouble, because they can cause damage by releasing harmful chemicals, using up resources, and even hijacking the cell itself. So in summary, a microbe is simply a tiny living thing, or a tiny biological agent that's too small to see without a microscope. Some microbes are harmless, some are helpful actually, and some can cause disease. But the ones we concern ourselves with here are the ones that cause disease. For our purposes, there are four main types of microbes you'll hear about. Bacteria, which are tiny single-celled organisms. Viruses, which are genetic material in a protective shell. And then we have fungi, which are organisms like yeast and mold. Then we have parasites, which are organisms that live in or on us and feed off the host. This is important because each one of these microbes has its own targets when it comes to treating them, so we use different medications, and it's important to know what we're treating so we can use the right medication. There are two main types of cells. This is going back to high school biology. We have prokaryotic cells, which are simple cells with no nucleus, and then we have eukaryotic cells, which are more complex cells with a nucleus, like our cells, for example. Bacteria is an example of prokaryotic cells. They have no nucleus, but they are full cells, just very small and very simple ones. They don't have a neat little DNA office, which we call a nucleus. Their DNA sits in the main part of the cell. They also don't have the same internal compartments that humans have. Human, fungi, and parasites, on the other hand, are eukaryotic cells. Think prokaryotic, but just a lot fancier. Their DNA is stored inside a nucleus. They have lots of internal compartments, which we call organelles, and these do specialized jobs. Viruses, on the other hand, are different again. They're not cells at all. No nucleus, no organelles, and no metabolism. They're basically genetic instructions in a protective shell, and they only do anything once they get inside of a cell. So, one of the first concepts we'll discuss in microbiology is staining. So gram-positive and gram-negative is just shorthand for the cell envelope, meaning what's around the cell. For gram-positive, the envelope is a thick peptidoglycan wall, that word will become important, that is outside the membrane. Peptidoglycan is a tough mesh-like material that makes up the cell wall of bacteria. Basically, the bacteria's armor that helps keep its shape and stop it bursting from water pressure. In slightly more detail, it's made up of sugar chains that cross-link by short peptides, peptides being little tiny protein bits. So in total, just think of it like netting reinforced with knots. Now we turn to gram-negative. These have a thin peptidoglycan wall plus an outer membrane. So the outer membrane itself contains what we call LPS. LPS stands for lipopolysaccharide. Think of LPS as a built-in outer coating made of part fat and part sugar sitting on the surface of that outer membrane. Your immune system treats LPS like a giant red flag whenever it sees it. The body senses LPS, it triggers a big inflammatory alarm response. The alarm response is helpful if it's controlled, but if it's excessive, it can lead to a sepsis type response. Sepsis type responses involving widespread inflammation, leaky blood vessels, low blood pressure, organ stress, etc., etc., etc. Now, the easiest mental model that I can think of to remember this and summarize it quite well is gram positive equals a thick wall no outer coat. Remember, they both have walls, but the gram-positive is a thick wall, no outer coat. Now for the gram-negative, it's a thin wall, but in place of that, it has an extra coat, which we call the LPS. So remember that gram-negative, it's a thin wall, but has an extra coat, the LPS. So to summarize, gram-positive have a thick peptide or glycan wall that holds the purple stain. That's why they stain purple. While gram-negative have a thin peptide or glycan layer, plus they have an outer membrane that blocks many antibiotics and also contains LPS, endotoxins. And we'll get back to this word endotoxin later. So the LPS outer structure can drive a strong inflammatory response, a sepsis type response. That extra coat, the LPS, is why gram-negative bacteria can be harder to treat and can be nasty in bloodstream infections. But gram-negative bacteria are not automatically more serious infections. The severity of the infection will depend on the organism itself. Now let's talk about oxygen. You've probably heard the term aerobic and anaerobic bacteria. Aerobic means that bacteria uses oxygen to make energy, so it grows best when oxygen is around. Anaerobic means it doesn't use oxygen, and for some bacteria, oxygen can actually harm or even kill them. This is useful information about the bacteria because it tells you where the infection might be. If there's lots of oxygen, like in lungs, skin surfaces, it's more likely to be aerobic bacteria. If it's low oxygen areas like deep wounds, abscesses like the pockets of an abscess, or deep tissue, gums, the bowel, or diabetic foot infections, it's more often going to be anaerobic bacteria. It can also guide antibiotic choices. For example, some antibiotics cover aerobes well, but not anaerobic bacteria, and vice versa. Another thing to note is smell and gas. Anaerobic infections can be foul-smelling and sometimes produce gas in tissue. The next thing to discuss in microbiology is bacterial shapes. You might hear terms like cocci and bacilli. These are the two main shapes bacteria take. Cocci being round and bacilli being rod-shaped bacteria. There are other shapes too, but they tend to be a mixture or in and around these two main shapes. We have cocobacilli, which are short, fat rods, so they're in between rods and being round. And then we have vibrios, which are comma-shaped or curved rods. Then we have spirilla or spirochetes. Spirilla are corkscrew-shaped, spirochetes are thinner and more flexible. Then we have filamentous or branching. These are long threads of branching rods. Some look a bit like fungus. And then we have pleomorphic. Now these are bacteria which have no fixed shape, so they can change from one shape to the next shape. Now these shapes often exist in groups and not individually. The main groups that they like to form when they lump together are staph and strep. Staph is when they clump together in a shape that looks like a bunch of grapes. We call this clustering. On the other hand, strep is when they form chains like beads. So for example, when we have staphylococci, these are clusters which are round in shape. And then we have streptococci, which are round in shape, but they form chains. So chains of round shaped bacteria. Great, now let's talk about virulence factors. These are traits that microbes have that help them to cause disease or do maximum damage. There are 4 particular traits that I want to speak on. Number 1 is having a capsule around the microbe. The capsule can hide its identity tags known as antigens, making it harder for your white blood cells, your phagocytes, especially your neutrophils, to find the microbe and eat it. The capsule helps the microbe to stick to the mucosa, letting it hide and have more time to replicate and grow. So basically, the capsule helps the bacteria survive that very first line of defense. Secondly are toxins. There are broadly 2 types. We have exotoxins. These are toxins that exit the bacteria, hence exo. They go out and cause tissue damage, leading to symptoms like diarrhea, vomiting, and nerve effects. Then there are endotoxins, which are on the bacteria itself. Once they are detected, they can cause an immune response, leading to inflammation and fever. And now the third trait is a biofilm. It's exactly what it sounds like. Sometimes bacteria stick to surfaces and then they have a slimy film literally cover them, making it harder to get to them and harder to get rid of them. They can then fire off infections from this biofilm, acting like a reservoir of infection. This happens in prosthetic valves or joints, chronic wounds, and even dental plaque. And lastly, we have spores. Some bacteria have developed the ability to shut themselves down and stop growing and basically go into what is like a pseudo hibernation mode. So when the environment around the bacteria is tough, they do this to protect themselves, but they can later wake up, which we call germinating. The spore form itself is a way for them to preserve themselves. Not all bacteria can do this, but some can. Examples of bacteria that can are gram-positive rods like Bacillus or Clostridium. Next, let's touch on viruses. Viruses are not cells, as we discussed. They are genetic material, DNA or RNA, inside of a protein coat. Sometimes they may have an envelope. When you have a viral infection, you can experience fever and myalgia, which is just the feeling of being sick. Now, a few other important terms to note with viruses are incubation period, which is the time from exposure to a virus to the time that you start to experience symptoms. Then there's viral shedding, which is the period where the virus is being released from the body. Shedding makes you contagious. So if a virus is leaving your body in secretions, it can enter someone else's body via the mucosa. Mucosa referring to the nasal, the mouth, or the eyes, or even other routes. You can sometimes shed before you even feel ill, depending on the virus. Next, we should touch on fungi. Fungi are eukaryotes. That means they have a nucleus, they have organelles, and they exist in two main common ways. You have yeast, which are single-celled fungi. Often these cause thrush-type presentations. And then you have molds, which are filamentous, and these can be invasive in vulnerable patients. But you also have dimorphic fungi, which can switch from one form to the other. Fungal infections often happen in specific environments, usually warm, moist environments, which allow overgrowth, for example, skin folds and feet, when normal flora balance is disrupted, or when the immune system itself is compromised. The last type of microbe we want to look at is parasites. Parasites are also eukaryotes and are often split into two. We have protozoa, which are single-celled parasites. They live and multiply inside of a host. They often cause diarrhea disease or systemic infection, depending on the organism itself. And the transmission is often through water, food, or other vectors. Then we have helminths, which are perhaps one of the most scarier ones to think about. These are worms. These are multicellular organisms, so they're not single cellular. They have multiple cells and they have life cycles, often starting from eggs to larvae and then in their adult form. The disease can come from adult worms living in the gut, larvae migrating through tissue, or your own immune reaction to the parasite itself. Ectoparasites, these are the parasites that live on the skin, for example, lice, scabies. The symptoms are often driven by your own immune response to them. Lastly, before we finish, I just want to touch on how we identify and do tests properly. There's a few key terms that you need to be aware of. The first is culture. This is growing the organism from a specimen. For example, you take a urine sample, blood sample, or even a swab. Then you begin to grow the specimen in order that you can identify it, and this can take some time. We then use microscopy, which is looking for organisms or immune cells directly in the sample by use of a microscope. And then we have PCR, which stands for polymerase chain reaction. This is a molecular test that amplifies the genetic material because it's so tiny, it's hard to detect. So we amplify it to make it easier to detect. Many of us are aware of this due to the recent pandemic with COVID. Many of us have done PCR tests before. For RNA viruses, though, labs often use RT-PCR, which is reverse transcriptase polymerase chain reaction. PCR is often fast and sensitive, but it detects genetic material which can persist even when someone is actually no longer infected. Then we have antigen tests. These detect specific parts of the organism, not DNA, but specific proteins on the organism. It's often faster and sometimes it's less sensitive than PCR. And the other way is serology, which is looking at your own antibodies. If you've been to war with a particular infection, the tools that you use in that war, which are the antibodies, we can use this to kind of tell the story backwards as to what type of infection you had or you were fighting. It's useful for past exposure or later stages, not really useful in early diagnosis. So that's it. And I hope you guys are now equipped and ready to dive into infections and anti-infectives.